Hi everyone, welcome to section 2. In this section we're going to use the biopax language as an example of an ontology which is used to represent biological pathway knowledge, and I'm hoping to demonstrate that you can use this to actually reason about biological systems, but also combine it with omics data to actually reason about what could go wrong with biological systems. You would have already seen with David the gene ontology, which is an ontology which is used to represent protein or gene product function, and it's split into three parts. There's the molecular function, which is the exact biological function of that molecule. So for instance, in the insulin pathways we saw glucose kinase, which was adding a phosphate to glucose to produce glucose 6-phosphate, which was the start of energy metabolism. So that's the biological, that's a molecular function, and then the biological process of that glucose kinase is actually energy production. It's a more general process that is involved in, and then the cellular components, it's actually an ontology of all of the different parts of the cell where these functions may be taking place. So that's the gene ontology, and it allows you to reason because it's a hierarchy. So you can say what functions are common, which ones are share a common parent, which ones are involved in the same processes. The biopax language uses very similar ideas, but to represent biological pathways, so they can be reasoned about. It uses the OWL ontology language, which is a W3XML standard developed for the semantic web. I should disappear at this point. In addition to the biopax ontology, there's another standard, the Systems Biology Graphical Notation, SBGN, which is a graphical notation that allows us to visualize the pathway information contained in biopax. So we'll see that. So this shows a class hierarchy of those classes which are involved in the biopax language. At the top of the hierarchy we have the entity object, entity class, and one particular type of entity is the pathway. So pathways are the main objects that the biopax is describing. How does it describe a pathway? Well, it represents it as a number of entities which interact via different types of interaction. Entities that include genes, but also physical entities such as proteins, DNA, RNA, complexes and small molecules. How do these different components interact? Well, they can interact via genetic interactions with genes, molecular interactions, so it might be small molecules interacting, conversions, for instance a biochemical reaction, so that might be one small molecule converting to another small molecule via an enzyme, so acted upon by a protein essentially. But there's also control mechanisms, modulation, catalysis. So that's the general class hierarchy and the different things making up the ontology. Each of these, like the protein, has a number of properties. So proteins, they have a name, they have a cross-reference potentially to a database. If it's a protein, that might be the UniProt entry for that particular protein. They have features of the protein, for instance, phosphorylation sites, or they might not be phosphorylated, so they have a not feature. They might have cellular location. So these are, each of the classes has a description of its properties. I think it's good to show a real concrete example because it's quite abstract. So this is an example which actually arose in the insulin pathway, which you might not sort of recall in lecture 1, but it's also involved in cancer when AKT is it's an oncogene which become unregulated and cause cancer, or some types of cancer. So, and it's responsible for cellular growth ultimately. So it's a biological process which is important. So AKT is a protein, and it's a signaling molecule. So there are other proteins, other enzymes which phosphorylate it. So PDK1 can phosphorylate AKT1 and add a phosphate to threonine 308, which is one of the amino acids in the protein. There's another enzyme which can add another phosphate to serine 473, so it becomes double phosphorylated, and in this state it's activated and it can combine to other proteins such as HSP90. And then there are enzymes that take it back again, that dephosphorylate it and dephosphorylate it back to an inactive state. So this whole network, so you could represent this with network interactions between sort of edges and nodes, but it can also be represented more flexibly in terms of the ontology. So our AKT1 is a protein reference. It has the standard name AKT1, so it's this one, has alternative name PKB, and it has a cross-reference to UniProt P31749. So this is a UniProt accession code, and David would have made you familiar with UniProt. How to describe the phosphorylation? Well, P at 308 is a modification feature. It has feature location AKT1 308, has modification type phosphorylation. So this is basically how the ontology works. It has declarative statements about components in the pathway, but also reactions in the pathway. And it tries to describe all of the different parts of the pathway and how they work. So this shows the part of the AKT1 pathway in SBGN, so a graphical notation. And you can see it starts with AKT1 with no phosphates. This shows a catalytic reaction by this enzyme, and that one will phosphorylate it to get a phosphate at 308. This is a second enzyme that when phosphorylates it to double phosphate, whereupon it joins together with HSP90. PP2A is another enzyme which dephosphorylates this, and it also dephosphorylates this one. How is this all described? Well, AKT11 is a protein. It has protein reference AKT1, has not feature phosphate at 308, has not feature phosphate at 473. Describes that. This is a reaction, biochemical reaction. Has left AKT12, so it's this one, AKT 1.2. It's a protein, has this reference, so it's the same protein as here, But in this case, it has the feature phosphate at 308. So it's got a phosphate here. And this biochemical reaction goes from here, AKT 1.2 to AKT 1.1, so this one, and it's left to right, the reaction. This shows catalysis by this enzyme. So catalysis 1 is catalysis controlled by PP2A1, which is a reference to that protein, has controlled reaction 1, has direction irreversible, so it cannot reverse left to right. So this is how the ontology describes the different individual parts. This is the assembly of a complex assembly between HSP90 and AKT 1.3, which is this double phosphorylated AKT. Has right complex 1. Complex 1 is here. It's a complex. It's reversible. So it can go into the complex, the two of them joining together, or they can separate because it's reversible. This is a description of the complex and all of the parts of the complex. So you can sort of see how a biological pathway may be described in terms of individual ontological features of that pathway. It's how we represent the knowledge. We think about this protein and the same protein with a phosphate and the same protein with two phosphates and the enzyme that turns it from here to here. So it's how we store this information ourselves in a way. So how would we use such pathway knowledge to help integrate omics data and get an understanding of a system and possibly how a biological system has gone wrong? Well, if we use a radio analogy, we have a network diagram, an electronic circuit diagram of all of the components. So this is an old transistor radio. You've got resistors, capacitors, and this actually tells you how all the components are joined together. The analogy to omics data, high throughput data, would be measuring all the voltages throughout the circuit between a working radio and a broken one. So that's essentially overlaying relevant high throughput data and voltages to try to pinpoint faulty components in the circuit. So if you measured the voltages throughout this circuit and you compared the working radio with a broken one, you would notice that voltages change at certain places, and this potentially would highlight maybe this resistor here is burnt out. It would actually pinpoint parts of the circuit that potentially are faulty, particularly as we have a clear mechanistic understanding of how electronics works. And that analogy, in a sense, is what we want to use for understanding networks, biological networks, for instance in cancer, by having an ontological representation of the pathway, and then proteomics data, or in this case potentially gene expression data, is easier to obtain as a rough proxy for enzyme levels. So we would get enzyme levels for these different enzymes, and then proteomics data for different levels of the phosphorylated AKT1, and potentially also the complexes that may be existing. By formally structuring this knowledge, it allows essentially reasoning. For example, if this enzyme decreases in concentration, then what's its effects? Well, this enzyme changes this double phosphorylated into this single phosphorylated, and it also helps catalyze the conversion of this single phosphate AKT1 to an unphosphorylated AKT1. So if the enzyme decreased in concentration, these pathways would become less, and that would actually result in the direction possibly being this direction, with more of this, which potentially could lead to cancer, because this is the signalling molecule for cell growth. It will also stop this pathway direction, so the complete unphosphorylated form of AKT1 would become lower, assuming that there are not other interactions within this pathway. You could treat it as a single entity. So you might see this as a vital enzyme, which if it was decreased in concentration, or if the gene was knocked out, the enzymatic activity by mutations, it potentially could turn on this pathway and cause cancer. And it's that sort of, because the pathways are now thousands of molecules and it's difficult to do human reasoning on them, particularly with the large amounts of omics data as well. It's that sort of automatic reasoning that would be useful to be carried out. So in summary, I hope to have demonstrated that to actually properly use microarray data or any omics data requires a substantial understanding of how the experiment was conducted, with understanding of the metadata that's required to be recorded. For instance, did the experiment use a spotted array? If that's the case, then it's 2 channels of data. We need to know something about the experimental design. Was it a pairwise direct comparison between samples, or were all samples compared to a reference sample? If it used Affymetrix gene chip, then it's a single channel device. So each sample would have been measured by itself without any reference. What were the experimental conditions or factors? If it's a human patient data, there are factors such as the age, which may affect gene expression data, the gender of the patients. All of these factors come into an analysis of this gene expression data. What are the sample annotations? What are the gene annotations? There's even more details in terms of the layout of the genes on the array itself, but this is really only required if you're going to analyse the structure of the raw data coming off the array. MGED Society introduced a number of standards to help with this standardization of microarray data. The Miami standard, the MAJO-M, MAJO-ML, MEG, TAB, MGED ontology. The key thing here was to standardize what researchers were actually saying and represent that data, so that it can be reused by different researchers and so that it could be transferred from one system to another and fully understood in terms of MGED ontology, for instance, defining precisely what terminology was meaning. We also introduced a second example, the BioPack standard for ontology for network biology representation. I think this is a good sort of example of where this declarative ontological representation can describe complex networks by just describing all of the parts and how they interact. And then you could reason about this by then overlaying it with omics data. So all of these standards are required. At the moment, a lot of machine learning is carried out by just using floating point numbers and not knowing what the genes do, and only knowing potentially one label of the sample. Is it a positive or negative sample in terms of, say, cancer? But this is very limited, and really a much more semantically rich understanding of the biological data would allow much more sophisticated machine processing and inference. Okay, so that was all about data representation in biology, in particular omics data and biological networks. Next lecture, we're going to actually look at applying some machine learning techniques to all of this data. Okay, I'll see you then.